Okay, very good. So, very happy to introduce Jérôme Chave, who works in the Center for Research on Biodiversity and the Environment at the CNRS of Toulouse. And he is one of the principal investigators for the SynTreeSys Cesab working group. His research fields are plant functional traits, biogeochemical cycling, macroevolutionary patterns, mainly in tropical forests. But he's involved in many different projects that involve remote sensing of tropical forests and map carbon stores globally. He works also on biomass and tropical tree allometry. He developed methods to infer parameters for Hubble's neutral theory of biodiversity. And he's also involved in metabarcoding and environmental DNA biodiversity surveys. And we also have Renato Lima, who works at the Laboratory of Ecology and Forest Restoration at the University of Sao Paulo in Brazil. And he is also one of the investigators of principal investigator of SynTreeSys, started as a postdoc and then, that evolved. And his research fields are also plant community ecology, theoretical and quantitative ecology, biodiversity, data management, and his focus is on tropical and subtropical forests. He also works on forest succession, dynamics, restoration, and regeneration, and is interested in plant functional trait measurement, collection, and imputation. So thanks again, everybody, for being here. And Jérôme, the floor is yours. Thanks very much.
Thank you very much, Camille. And I actually will leave the floor to Renato, and we'll be moving the slides forward. Renato.
Yeah, so hi everyone. Thanks for joining us. It's a pleasure to present some of the results and ongoing progress of the SynTreeSys group. We're going to share the floor, me and Jerome presenting. I will present half of the results and what we've done so far, and then Jerome will finish it off. Just this introductory slide to tell you guys we are working with 3 species and we are working in Latin America. So we are talking about the region with the most biodiversity in the world related to trees and many other groups as well, other groups of organisms. So the tropical America is one of the most diverse places on Earth, and we need lots of information to help to understand and describe the patterns on that region. So we gather lots of information in SynTreeSys. That's one of the goals, using plot inventories and not only occurrence data. And this is quite important because plot inventory information carries not only occurrence information, but also the abundance of the species in those sites as well. Next slide, Jerome, please. So we are really nice and diverse group of people from many different countries. From the get-go, we try to involve as many people from as many networks, plot inventory networks that we had. We're going to present those networks later. But we have lots of people from Latin America as well. So this is one of the things we wanted to do from the start, having representatives from as many countries as we could. So a really nice group of people with different backgrounds. And it was a really nice experience to be those 2-3 years with everyone. Next slide. And we had 6 meetings. We started in 2022. We have 4 of them in the very nice building of Cesabi. They were super nicely organized. And we have 4 of them in Montpellier. Some of the pictures, probably other groups from Cesabi as well know some of those places that we go while being in Montpellier. It was super nice. Next, please. And we discussed, and Cesabili gracefully granted us with the possibility of doing 2 meetings actually in the place we are working with, which was Latin America. So we have one meeting in Brazil in December 2024. It was hosted here in my lab. It was super pleasure to host one of the Cesabi meetings here. And we also had a meeting hosted by the Humboldt Institute in Colombia It was really amazing. We have some pictures of us there, and it was quite important as well to get in contact with local researchers and local actors. We did. We're going to show later some actions to get involved with the local communities in those places. And we are super grateful to Cesabi as well for allowing us to do two of our meetings outside Montpellier. Next. As we said, we are a network of networks, more or less what we're calling now a meta network. It was really a big challenge to get everyone together, get information with different formats together. But we are super important to achieve the results we have so far. So we have Treeco database, forest plots, the Amazon tree diversity networks, the Caribbean forest more based on the Caribbeans, the dry forest, which are more focused on the dry environments of South America. We have Bosques Andinos groups as well, which is more focused on the Andes. And we have lots of other networks involved. Guia4 from the Guianas, BioTreeNet, mostly based on Central America. And also Monafor based on Mexico, Panama as well, people working in the Smithsonian Institutes there, and other smaller groups that actually start to joining us as we went forward with the work. So it was really nice to put all this information together, and it was also a challenge. I think we're going to present this in the next slide. How to put all this information together was actually a big challenge, but it was super nice challenge. Next, please, Jerome. So we're talking about compiling information from 31 different countries. We achieved a total of 867 collaborators throughout all those networks I just described you guys. Over 13,000 plots, over 14,000 accept name species in the World Flora Online, and we are above almost 6 million trees, combining all those inventories from all those plot inventory networks. We have these live maps kindly prepared by Nicolas Casajus, which is available for us to fly by, and it shows the location of each plot of each network. So there's a legend there. So it's interactive. It's quite nice as well. Thank you, Nicolas Casajus, if you're there somewhere. Next, please, Jerome. This is the main summary of the information we gather from all the networks. We have the totals on top, but we have more or less what each of those networks I presented you guys provided in numbers of plots, in plot area, numbers of trees, and number of plots observations, so how many species per plot, so numbers of records, abundance records actually. There's also numbers of species there. Those are not yet clean number of a species, so we have this high number of 40,000 possible species names from all of the networks. So this is pre-taxonomic homogenization. And those are the numbers of people from all the networks we gather. There are some networks that have really a large number of peoples. They have different number of different rules as well in terms of data access. So we try to adapt to the realities, to the size, and to the data sharing rules and agreements from all of those networks as well. Some of them are more open access. Some of them are more under. You need to ask for the data. You need to request collaborator per collaborator. So it's they are quite different, and they contributed a lot for different regions of Latin America. Next slide. Thank you, Jerome. So this is the outline of what we're doing in terms of science in SynTreeSys. This is not the exactly work packages we anticipated in the original project. We changed a little bit. Some of the work packages were not started yet, and some other ideas came in along, and we are working on them right now. So there are some work packages which are not mentioned here, but they are ongoing in a sense as well. So we have one main work package working with tree diversity. So the focus here is just to describing the major patterns of tree diversity across Latin America and describing the similarities and differences within biomes, especially. We're going to show those results later. Another group, core group of our group, working with alpha diversity, just trying to explain why there's this number of species in one region and less species in the other region. Tree vulnerability, which is how individual species are threatened due to changes in climate and land use changes. So then is more like more related to species distribution modelings. And we have two more or less opinion papers that emerged of all the work we had trying to work together and combine all this information. We have one paper about some simple rules on how to manage taxonomy and ecological data sets, and another perspective paper on how to work on meta networks as we did in SynTreeSys. We're going to present one by one now. Next one, please, Jerome. So the first work package, in a way, is what we call it the tree diversity work package. We are focusing on dividing the Latin America in different biomes. We are using the IUCN ecosystem functional group topology to describe those biomes. It's not exactly the classic biome definition. They have kind of a functional component to it. So it divides what we call generally tropical rainforest into smaller biomes as well, like flooded forests or heath forests as well. But important from those slides here is to show for those not acquainted to Latin America is that Latin America is not only the Amazon, is not only rainforests. So we have dry forests, we have savanna spread all over Latin America as well in different spots. We have dry forests, a large contribution of dry forests in terms of area and in our plots as well. We have mountain forests, cold forests, like subtropical temperate forests in South Brazil, but also in Central America and in the Andes as well. So there's a big diversity of biomes in Latin America as well. Next one, Jerome, please. So these are the plot distribution. So these are the colors of the plots we have, the 11,000 plots we have in our data sets colored by biomes. We assign those biomes based on vegetation types, if available. If not available, we have this standardization that we try to assign to every single plot the biome they belong to based on species composition or most abundant species. So we have the distribution of those plots and the biomes to the right here in the slide. Next one, please, Jerome. And this paper is really trying to provide the big numbers and descriptions on our data set for each of those biomes. So we have there, every row is a different biomes, and we are describing in this table how many number of individuals we have in our data sets, number of species, total area. And to the right, which is important here, is that the first result of what we are seeing with this data set is that we really have a low sampling coverage for tropical America. It's really one order of magnitude what we have generally in North America and in Europe. So we really need more plots in the region, particularly in mountain forests. We really have a small sampling coverage. Next one, Jerome. So we are talking about the biome distribution in previous slides, and here is important result as well. The lowland rainforests, like the Amazon and other rainforests we have in tropical America, they are super important in terms of number of species and number of unique species as well. But tropical America is not only about rainforests, it's not only about the Amazon. So other biomes combined can have as many species as the rainforests. So we have a big contribution from dry forests, mountain forests, and other biomes in Latin America that combined, they can host as many species as the rainforests. And these have really important implications in terms of species conservation in the region. Next slide, please, Jerome. We also fitted some species abundance distribution. Well, we fit neutral models to the species abundance distributions for each biome. We can see here the main biomes we are considering for analysis. Almost many of them didn't really have a really nice fit in the first most abundant species. We call them ultra dominant. So we have the 5 to 10 species that are really abundant, and they are not fitted well by a neutral theory model for the species abundance distribution. But they actually describe them quite well, to the point that we can actually compare them and actually estimate the probable number of species that we would have in each one of those biomes. Next slide, please, Jerome. So these are the numbers of species projected for each of those biomes. We have the Ewens theta from the neutral theory model fit for each biome. And we have the total number of species projected for each biome and for the entire region. So we can see here to the extreme right in red, what we call the estimated number of species. We probably have viable populations in tropical America. We are defining those viable populations of those having predicted population sizes above 50 individuals, adult individuals. But we are talking about, in total, having 26,000 tree species in tropical America, which is at least 20% more than other predictions we had for the region so far. Next slide, please, Jerome. So a different work package now is not only about big numbers and big numbers by biome, number of species, is more describing alpha diversity. So what is the explanation for the number of species per plot? And here we are trying to explain alpha diversity actually in terms of climate variables, but also in terms of the biomes itself. So if the biomes have different relationships between alpha diversity and climate, but not only climate, I'm going to show you guys a little quicker, real quick, the models is not only climate. And if there's any different relationships between those environmental variables and the biomes. So really now we are talking about how many species we have per plot, per environment, and trying to explain why we have more or less species in each of those plots. Next slide, please, Jerome. So here is the plot of our coverage, the plot distribution of our data sets in the climate space for tropical America. We have here mostly 4 climate variables, which is annual temperature, annual precipitation, vapor pressure deficit, and water deficit, which gives us a bit of information of rainfall seasonality. So we have temperature, precipitation, and seasonality there. And this is an image of how our plots are distributed in this climate space. Next one, please, Jerome. This is the model we are applying. As you guys can see here to the right, we have all those variables and interactions between variables trying to explain why we have more or less species in one of those plots. But we also included there other information such as methodological information related to the minimum DBH cutoff of each plot, the sampling coverage, the plot size as well. So we're calling those as sampling effects on the species richness, which are quite important. And for both work packages we mentioned, we have some filtering, pre-filtering of the database that we need to have the minimum information available for each plot to compose the analysis. Won't go really further into details here. Next slide, please, Jerome. So these are the main results of the models we have so far, and if they are significant or not for explaining alpha diversity in Latin America. We're going to leave it there for consultation later. I won't go through each and every result here. Next slide, please, Jerome. And here is a nice representation as well of each of those biomes, which we're using as a grouping variable in our analysis in the climate space as well. So we can see that rainforests, they really have more annual precipitation and more annual temperature than montane forests or temperate woodlands, which are more in Central America, where we have more temperate component for species compositions as well. And then we have the dry forests, really different in terms of precipitation from the rainforest. So this is the climate space for each of those biomes in our data set. Next one, please, Jerome. And here we're trying to explain if there was any difference in those alpha diversity, climate or environment relationships across biomes. And we can see that for some of the biomes, we have positive relationships with some of the climate variables. For other biomes, we have negative relationships or negative slopes for those relationships. So yes, we asked in the previous slide if there was any differences between the relationship of alpha diversity and climate variables for explaining tree diversity, and the answer is actually yes, and it's quite variable across the climate covariables we used in the models. Next slide as well, Jerome. So when we think about species diversity and particularly tree diversity in Latin America, we always think of Amazon, and we actually could confirm that Western Amazon has a really large number of species, but not only in the Amazon, right? So we have this really high tree species diversity elsewhere as well, importantly in the Choco region, Mesoamerica as well, and some stretches in the East and South America in the Atlantic rainforest that we could have a really high number of alpha diversity outside the Amazon. So again, here the message is like when you think of high alpha diversity across Latin America, we are not only talking about the Amazon as well. Next slide, please, Jerome. And this is, I think, the point where Jerome takes the floor and finishes off the presentation. Thanks. Thanks, Jerome.
Thank you, Renato. Thank you very much. So yeah, carrying on, the third topic we would like to cover today, and we have covered as part of the SynTreeSys meetings, is study of the vulnerability of tree species. So as you've seen, we've been cranking up big numbers. How many species do we have in tropical America? And we came up with 26,000 species, and we broke it down by biome, which is novel. No one had done this before and with this magnitude. The second result we had, as Renato just showed, was exploring alpha diversity, local diversity across the different environments that we have in Latin America. And the third result is boiling it down to the species level and trying to address how species independently from each other are distributed across this environmental gradients, which is a very classic question, I suppose, for CESAB participants. So understanding the distribution of species is also understanding the vulnerability of tropical forest species to a number of threats, polycrises. One of them is the climate and biodiversity crisis. Another one is knowledge gap, the lack of knowledge, basically, which makes documentation of these problems very difficult. The impact of climate change is one of them. And of course, land use change is probably the dominant cause of threats to biodiversity. Then this project is still ongoing because it's facing a very large challenge. But what we can say for the moment is that we aim to map the distribution of tree species across the entire range of tropical America by first investigating the spatial distribution of species composition using species distribution models. But also, we like to model the abundance of species using species abundance models, which is obviously much more complicated because presence absence data are first layer of information that is pretty well known. Next, we like to determine the climate sensitivity of tropical America's tree species, being one by one and across the range again. Some very important work, recent work by co-members of the SynTreeSys program have looked into the Amazon and looked into this question using Amazonian tree species, but we'd like to expand this beyond the Amazonian rainforest, bearing in mind that a lot of the species in the Amazon are also outside of the Amazon. So this is not a clear-cut border in terms of biogeography. And third, we like to examine how future changes in climate and land use change will affect species contribution, and so develop forecasting scenarios for this region based on climate change scenarios, but also land use change ones. So as a concrete example of what we have in mind here is that the southern border of the Amazon is much drier than the central Amazon and has a distinctive suite of tree species, which are disproportionately threatened by the deforestation trends today. So looking into these specific regions is very important, and only by looking beyond the Amazon into the drier forest zone, we can really assess the vulnerability and the conservation status of these species. Now, what we've been doing so far is more trying to model the abundance of species, so on a species by species basis using bioclimatic variables and soil variables, with pretty much classic approaches by now. And model distribution of species as well, using the same environmental variables. These are met with this framework. This is pretty much the same type of presentation that I repeat here. The top here models the abundance of species based on a number of environmental variables, species abundance models, and then mapping this species abundance model onto a geographical extent of interest. And the distribution is a distinctive project. As you may know, it's not easy to translate species abundance model into species distribution models and the other way around. So it's important to have those two streams of analysis separate for the moment to finally map the species abundances. A concrete example is the species Xanthoxylum redionanum, which is a dry forest species, rather fast-growing species in the Rutaceae. A very nice trunk and distinctive trunk spikes. So it's occurring all across from Central America to the southern part of South America. And so it's a good example with which we can test our case. And the end result here is the distribution of these species from the Yucatan Peninsula in Mexico all the way to southern Brazil and northern Argentina. Now, these type of results are still ongoing. This is part of Guilherme Grit's PhD project, and hopefully we'll be able to move this forward in the next couple of years. But this is very much work in progress. I leave you for this project with this photograph we took at Cesab with all sorts of nice diagrams to make sense of the raw data all the way to the right with extinction, the risks, and this type of metrics that we'd like to extract from this data analysis pipeline. Now, moving on, we also were faced with a massive challenge of standardizing the taxonomy of almost 6 million trees, each one of which was identified in the field by different teams over the span of maybe 60, 70 years. And so trying to standardize is a very massive undertaking, one that SynTreeSys has been faced with, but of course, Cesab groups are often faced with this challenge. So we came up with our 10 simple rules for this type of challenge, but really aimed at plant taxonomy identification for ecological applications. Of course, identify your individuals is key, but keep the records and verify your identification. Follow taxonomic backbone. And the one we followed is the Wildflower Online for reasons I'll soon explain. Store full species name, but also including author names because some other variants cause problems times to times. Use correct taxonomic terminology. Don't confuse them. CF and F and other taxonomic shorthands names. Deal properly with incomplete identification. Keep track of identification changes through time. So this is really a plea for making sure that we attribute the cause of changes in data sets, and we're able to backtrack if there is an error. Use name matching tools carefully. For this work, we worked with a Wildflower Online, for which there is a R package available called Wildflora, I think, and it caused quite a bit of problems because the name matching fuzzy algorithm is not perfect, to say the least. So we needed to go back and to verify the tree species one by one, and with 26,000 species names, it's not a small feat. And provide as rich metadata as possible. So an example of a species we're faced with is Inga thibaudiana, which obviously is a very beautiful tree species of the Amazon, but it looks quite different in the field than it looks in the herbarium. So combining knowledge is not trivial. And for this group of the genus Inga, which has over 300 species, this is a real challenge practically. Now, this is an ideal case where we have flowers, but most of the cases in the field, we have sterile specimen, and this is a major problem for our work. How do we make sure that we have identified the proper tree to the species? That's sometimes not just impossible or based on expert knowledge. I said a few words about the Wildflower Online. This is a very interesting new project, which is based on the expertise at the family level, plant family level, for a large number of experts. And this is moving forward quite quickly, meaning that every 6 months there is a new version of this database, and the version includes all sorts of synonyms for each species that is included in the collection. Now, with over 400,000 species of angiosperms in the world, this has become a necessary tool for solving the problems that we have for SynTreeSys. And we've been speaking a lot with Wildflower Online's leaders to make sure that we could also provide some useful feedback as a scientist from the ecology and evolution community. But much more difficult example is this one from the Sapotaceae Planchonella pachycarpa, which is actually noted in the literature. If you Google it online, you'll find some names. It's Goya-bio, and it's a plant. It's a timber species, so it's known as the timber species. But it's nowhere to be found in the taxonomic literature, and it ends up probably being a Chrysophyllum lucentifolium, which is known Sapotaceae tree species, but it's quite different. And it took quite a bit of searching, several minutes to several hours, to actually make sure that the species in our data set matches the correct tree species. Now, this type of example is not rare in our data set, and so this is why we need to be so careful at curating the data well. Now, moving forward, we also were faced with the experience of building not a network of experts, but meta networks of pre-existing networks. Renato told you that there's been decades in a work of collating plot data for asking a number of questions. The Amazon Tree Diversity Network focused on the Amazon was really a founding component of this project, but it's been going on for several decades now. The rainfall and forest plots have also been instrumental in asking questions about the ecology of tropical forests, and especially with very high quality and dynamic data, dynamic plot data. So trees that were measured not only once, but several times. TRICO, which is led by Renato, was also started to understand better what was going on in the Atlantic rainforest and this type of biome. And finally, but not only, the dry floor for the network was more interested in dry forest and environments. So bringing together all these different networks takes some understanding of what was their rules and how can we construct a project based on the smallest common denominator and common understanding, making sure that we don't trample on anyone pre-existing network, but empower them in the longer run. So it means understanding how to build a community, how to be inclusive, how to negotiate across networks and across scientists, how also they benefit from this work, and how we can enhance the impact. And we ended up collating information about that many plots because we have so many collaborators. But in itself, it's a challenge to even to submit a paper with 860 collaborators. So, and we have not done this yet, but please bear with us. It will be an interesting challenge in itself. Building trusted relationship is, of course, something that is crucial, and it's not trivial at all. Building a good reputation and trust is it takes time, and this is why I also think beyond the fact that we've been tackling a difficult problem, we've also taken a lot of time to build a community and to exchange. And this is also why we have not published any paper yet. And it's no excuse, of course, but this is some form of an explanation. We are trying to work slow but well, and hopefully this will pay off in the long run. By long run, I mean within the year 2026, hopefully we'll have some output. Don't worry. Fostering communication is key. We have had internal webinars within SynTreeSys collaborators. We've invited online a number of colleagues who were not able to attend the meetings in Montpellier or elsewhere, making sure that we updated them and they updated us with ideas and got their feedback. We also created a number of new newsletters internally across this large number of co-authors, so they were brought up to date and were aware of the progress. And we also engaged with local academic and conservation communities. We did this especially effectively in Colombia with the Humboldt Institute, and but hopefully collaboration with IPBS and other national hubs of these international initiatives will be very helpful into the future. And IUCN is another obvious entry point for several of our activities. One of them is to assess the conservation risks for each one of the species, as I said before. Also trying to do a better job at mapping biomes is essential for the IUCN, and this is something we are also willing and probably able to engage with. Now, this has been through a number of very nice discussions. This is a picture we took in Instituto Humboldt in Villa de Leyva, with a lot of very interesting discussion, intercultural and very open-minded. It's been a very nice experience. I think I'll speak on behalf of all the team members. We really enjoyed being part of this team, and it's been on a personal level and human level a very massive success, I would say. Now, looking at the conclusive couple of statements into the future, where do we stand and where are we going? SynTreeSys has established a fundamental milestone in the synthesis of tree diversity in the most biodiverse region of the world. That's an established fact. There's still the tip of the iceberg, many more legacy data, tree inventory data, and more in the drawers, in the gray literature, waiting for us to collect the data and improve our knowledge in this very important question. So we are still not yet there yet, and we'll make progress if we are allowed to keep on working together. Biodiversity monitoring is evolving, and new data will emerge. So it's not only collecting the data, the legacy data, it's also being aware that new data is being collected and knowing and including these partners in this exercise. Now, there is a need for a permanently funded infrastructure that would embody the vision of SynTreeSys. This infrastructure should not be in Europe or North America. It should be in tropical America and should be led by tropical American scientists. How it will be funded in the long run and how much security it will provide to the lead scientists and to the data themselves is to be decided and to be discussed. But for sure, CESAB has played a crucial role in this endeavor. Now, another very important point is that remote sensing has been instrumental already in specializing the data that we've acquired, but it's also making sweeping progress toward improved Earth observation products. And we need to be aware and pay attention to this progress. Finally, of course, deep learning modeling methods are shedding a new light on the research question. It creates a lot of opportunities. It creates obviously a lot of questions as well about how we can keep ownership. And by we as the close to 1,000 people that were cited several times already in this endeavors. So we need to be to think about the governance of these new generation of modeling tools just as much as the governance of the data. And finally, I would like to thank you on behalf of the team and repeat that none of this would have been possible without the generous funding of the Fondation pour la Recherche sur la Biodiversité and the CESAB, and especially the fantastic staff at CESAB, which has been so helpful. So thank you to all of you.
Well, thank you very much to you both for this very nice presentation. We're going to have a time for questions and an exchange time. So if anybody has a question, you should now be able to activate your microphone and your camera if you want to. It's nicer if you can. And if you can't do either, feel free to write your question in the chat and we will read them out loud for you. Does anyone have a question? Use the tool to raise your hand. That way it allows me to see you better and distribute the speech. And Jérôme, if you want to stop sharing your screen so that maybe we can see each other a little bit better, and I'll change the view to standard or gallery. Yeah, very good. Awesome. Thanks. Yeah, so maybe we can start with Claude Garcia. Are you able to? Yeah, very good.
Yep, I do have a microphone. Thank you, colleagues. So my name is Claude Garcia. I'm speaking from Zurich. I will not say how impressive the work you've presented is. It is. I have a question about the second part of the work where you were actually exploring the futures and how biodiversity will respond to that. I was involved a couple of years ago in a work a bit similar in Africa. I think you are probably aware of it. The paper was led by Maxime Réjoux-Méchant on unveiling African rainforest composition and vulnerability to global change. And I was wondering if there was something in the methods we developed to explore the impact of agriculture and roads, the impact of people on the forest biodiversity of Central Africa that could be of interest to you. Are you aware of this work? Have you connected with it, or is this a parallel line of study? And once again, congratulations, an amazing work you have presented.
Renato, do you want to answer?
Yeah, I've seen real quickly the study. I didn't really dig into the methods to know if we would have a similar approach or similar co-variables to address it the same way. I'm not sure we'll be able to reply this question
Okay.
to Claude. Maybe Adriani
So
raised her hand.
I can give my 2 cents. The Maxime's work is fantastic, and that's been a very nice project. The tree diversity in Africa is about half that of the Amazon, and we are spanning a much larger area, including Central America, thousand Brazil, Argentina, with very different dynamics in terms of development, in terms of encroachment of different types of population. So at that scale, exactly the same project would be difficult to undertake. But it would be very interesting to explore the methodologies and also ask whether we have simply enough data to explore this question. In my gut feeling is that it will be highly dependent on the futures scenario. And for the moment, the dynamics in Central Africa are quite different from the dynamics of South America. So contrasting the two will be fascinating, but also quite difficult. Thank you very much for an insightful question.
Adrienne, did you want to say something? Ask a question?
Yeah, just because I was more involved with that part of the project. I just want to say that the paper from Maxime is actually a big inspiration for us, and we are aware of the paper and we've been using it as an inspiration. Actually, Claire Fortunel is one of the colleagues in SynTreeSys, and she was also involved in that first paper, and we have been talking to her about similar methods as well. But as Jerome said, different system and also other complications. But yes, we have been using that as an inspiration for sure.
Brilliant. Thanks very much for your questions. Does anybody else want to ask something? I have a few questions in the meantime. If nobody wants to ask a question, then I can go for them. So yeah, I was very shortly involved in the problems that you encountered at some point with the taxonomic resolution, and I can testify on how complicated that was. And in the end, I just had a very methodological question for you, Renato or Jérôme, if you know. Like, do you have an estimation about the potential loss of, I don't know, trees that were inventoried, but that you weren't able to connect to a proper species name or something like that you had to discard maybe from your data set? Or what did you do with them in the end? Or what will you do?
Yeah, thanks for the question. Yeah, you were part of this process, and actually the decision making, what we did, we went like rogue in terms of trying to solve the problem. And as Jerome said, we went trying to manually check some of the most problematic issues. So it was more or less about 95% of the cases we could solve it automatically.
Wow,
we were confident of what that name represents, so where should we place it.
Oh, really impressive. I thought that would be much less than that. Okay, cool.
Yeah, there was more or less 5% that we need manual checking, and we are talking about the number of names we have from all networks compiled was about 45,000 names. So those are names with and without author, so all possible accepted names on the networks combined to get more or less a number of the 20 something thousand species valid, well, 14 actually in the plots, 14,000 valid names. But those 5% actually, we're talking about verifying more or less 2,000 names. So what we did was to prioritize those which were more abundant first, to check them first. The more problematic ones, the ones which could be names related to South America, or what we call the homonyms, so names which could represent two different species, the ones we were prioritizing. Some of the examples that Jerome said as well, some herbarium names, local forestry science names that does not exist anywhere. We try to solve them to make sure we don't lose records, we don't lose species as well. And we actually did it twice now already, what we call a checkathon. We just get together, we divide the work, everyone gets a chunk, and we start checking names and try to assign. Were we able to check it? Was it verified manually by synthesis team? What was the decision making? We managed to solve it or not? Should we consider it at the species level, genus level, if this is an index species? So we created a table and tried to prioritize that manual checking as well. It's still ongoing, and I think will be like hundreds of them that would go unchecked before the first paper. But at least we know which names we need to check, which are more important to check right now or not.
I see. Well, very impressive, and congratulations for that. I had another question, or actually just like a comment to start with. Like, I really appreciated the part of your presentation where you thank all your collaborators and you talk about how you were inclusive and how it was really important to build an effective network to make sure that the information was spread out to everybody and that they were involved. And so my question is, are there any reasons that were kind of universal among all your collaborators to, like, what was the benefit for them to be contributing and spending so much time and effort into trying to be part of this network? Like, there are some reasons that might sound obvious to us, but maybe there's other motivations that you might not have thought about in the first place that turned out to be important. Was that the case, or not really?
Well, I think so. I would not be super comfortable to speak about this alone, but I see Toby Pennington is here, and Oliver Phillips could weigh in here. I think the bottom line is that we are all scientists, all 860 scientists, and are all interested in big questions in science, and they're all really willing to discover this big picture. So I think this is the main driver. Of course, it comes at a cost, which is spending a lot of time in the field, organizing the infrastructure, and this should not be overlooked, obviously. But it's so easy to overlook this reality of going in the field and maintaining infrastructures and maintaining research groups in some challenging areas or environments. So I think the hope is that this type of exercise will go a little way towards helping those groups to get funded and get some support in the long run. But that's not a given, and there's no magic solution. There is no silver bullet in this work. We really need to keep on repeating the same obvious fact that this doesn't come for free. There is no free lunch here. We need to go back in the field and measure and monitor and do some careful botanical work, because without this, there is nothing in terms of biodiversity knowledge in this part of the world or elsewhere, by the way.
Of course. Renato, do you want to complete?
Just to complement what Jerome said, in more technical or practical aspects of it as well. We try always to adapt to the network realities and give them feedback as well. There was lots of data exchanges and data curation process. So for some of the networks we're working with, all those exchanges were really important. They have new versions, they included more data, they have new collaborators. So I think we tried as much as we could, not just to say, okay, give me your data, we're going to use it. It was really a two-way process in trying to improve at least the databases as well. And all those communications we try to provide drafts of the papers really early on to everyone, so they can really say, guys, I don't think this is a good way to follow. These are new ideas. So it is impossible to put the 867 people on a workshop, right? So we tried to communicate as much as we could and try to share early our ideas and results to make sure we could get their input as much as we could. And this comes with a cost. So it was hundreds of emails, hundreds of replies, emails in Portuguese, emails in Spanish, English, French, adapted replies to gender. So it was a big communication task as well. It comes with a cost. But I think most of our contributors and most of the CESAB groups, we are comfortable with what was done. We try to respect all the rules from each network. We follow them in terms of data sharing, data agreements, difference between networks. So we try to, data-wise and data sharing-wise and collaboration-wise, try to make it as healthy as possible. But it's almost impossible to give it back to all the 867 the same amount of effort they put into the field, as Jerome mentioned. But at least we tried to do as much as we could, and we're still doing it in a way. So it's fun.
All right, and thank you very much for doing so. And because this is going to be, and already is, a significant contribution to the biodiversity knowledge. So it's going to be awesome. And I guess it underlines as well the need, like you were saying, for like a permanent infrastructure and funding for these kind of initiatives. Awesome, and congratulations again. I guess if there aren't any other questions, we can stop here. Does anyone want to ask something or just comment? Still have a little bit of time. And otherwise,
There's some people
yeah.
in the chat.
Yeah. Thomas Couvreur. Textbook Cesab project indeed. How does your data relate to other similar efforts like Bien, led by Brian Enquist? Of course, it's a different level, but I think they also use plot data. Or could your data be fused with these initiatives?
Okay. Sorry.
Yeah.
No,
I think Bien has both herbarium data and plot data. I would say we have a great overlap in terms of plot data as well, related to Bien, but we never actually tried to see what was the amount of overlap we have with Bien. It's really nice question, nice idea. We should perhaps talk to them in the future to make sure we have the same information or not, and if they should get on board with us as well. Great idea. Thanks, Thomas.
Yeah, and also to complement, of course, this is not all data. It's not because we compile the data, it becomes all data. And I think it's the same for Brian Enquist's project. So it all boils down to whether we want to include more networks in this meta network, and under which terms, and what would be the advantage of doing this, what would be the spatial span and temporal span that we would include. So basically would hold on to asking what would be the mutual benefits of doing this. And I'm sure that Brian and us could discuss this. I know he's been involved in different Cesab meetings, so I think he's very much aware. So this is a conversation we can have in the future, maybe in the next generation of SynTreeSys with Cesab or elsewhere.
Yay, more collaborations. Awesome. Okay, I think we can call it a day. I will stop the recording.